My name is Connor Verkel, and I'm going to be talking to you about optical coherence imaging of microvascular oxygenation and hemodynamics. And really, that's just a complicated way of saying we want to do imaging of oxygen metabolism in vivo. And so I think we've already seen from a number of our talks today that metabolic imaging is a very hot topic in biomedical imaging. And it's a very valuable tool for studying biological responses throughout the body. And it can provide quantifiable measurements for the supply and demand of oxygen. It can provide important biomarkers for the presence or progression of disease. And so, because it's such an important topic, methods have already been developed using fMRI and PET to sort of perform metabolic imaging. But if you want to, and this is great for larger scale imaging, but if you want to look at the level of individual blood vessels, if you want to look at the oxygen saturation and the blood flow within individual vessels, you sort of need to move to a different modality. So that's why we use optical coherence tomography. And that gives us micron scale resolution. And it works in many of the same ways as ultrasound, except using light instead of sound. So for ultrasound, what you do is you send out a sound pulse and you measure how long it takes for it to be reflected back from different depths in tissue. In OCT, you're sending out light and you're measuring the interference pattern that's created from the reflections in tissue. And these reflections will change with depth. If you look at a shallow reflection, it's going to be much lower frequency than a deeper reflection. And so these frequency components let you tell where the depth, or how deep you are in the tissue. So in any given sample, you're actually going to have numerous reflections in the tissue. So your actual image is going to look like this. Luckily, we can use a Fourier transform to analyze the different frequency components and extract the different depths in the tissue where the reflections occur. So if you look up here, this is an image from a retina, and it just sort of highlights the structural imaging that you can get using OCT. In this case, you can see the different layers of the tissue, and it's just a good example of the high-resolution capabilities. So what makes our OCT system any different than the other OCT systems available? If you look at the schematic, it's pretty much exactly the same as any other Fourier domain system, except for one key difference, and that's that we use visible light source. Now, most OCT systems will use infrared light because they provide better penetration depth in tissue. But here, we want to trade a little bit of that penetration depth for more functional information that we think that the visible spectrum can highlight. So if we look at this, so hemoglobin is the component of blood that allows for oxygen transportation throughout the body. And it does this by binding to oxygen up to 4 molecules. And if you look at the light absorption spectra for oxygenated and deoxygenated hemoglobin, which is shown here, you can see that they're quite different, actually, especially in this visible range where we're looking. And we want to use these differences to help us quantify the oxygen saturation in any given vessel. And also, I should note that while a lot of optical systems want to pick this region for lower hemoglobin absorption, we actually want this higher absorption coefficient for better contrast in our images. So we can scan our OCT line along a tissue sample and acquire a structural image. But since we're looking at information that's contained in the blood, what we want to do is perform a technique called speckle variance. And what this does is if you take 2 samples separated by some small time delay of the same area of tissue, then what you can do is you can actually subtract out all of the static structure, leaving you with the dynamic scattering problem, or the dynamic scattering part. And this helps us visualize blood flow because the static scattering, which is just sort of the static tissue, isn't going to change between the two images. But the blood flow, the orientation and position of these different red blood cells is going to change with time. So the scattering pattern that you acquire inside the vessels is going to be different, and you're not going to subtract that out. So that's why you get this nice contrast of the blood flow. So if you took a 3D data set of this speckle variance, what you can actually do is you take a maximum intensity projection through that data set. This is looking from the top down in a rat brain. You can get a really nice high resolution angiogram. And I want to quantify this with, this is more on the preclinical side. So this is an invasive procedure for a rat. We have to go in and thin the skull so that we can actually get light through the skull and acquire these kinds of images. That said, it is less invasive than doing, for example, a craniotomy or a cranial window. So if we go back to the cross-sectional images of this dynamic scattering, we can see a couple different features. So you can see there's these little dots, and those are the blood vessels. And under some of these dots are these tails, these bright trails of dynamic scattering events. So when light comes in, there's a chance that it'll be back reflected, and that's where we measure these, the bright spots, the vessels. But there's also a chance that it'll be forward scattered when it hits the blood, and then back reflected from some depth in tissue below that. And it's this multiple scattering tail, this region that we think has a lot of information about the absorption of the blood within the vessel. So if we take a window under the vessel of this dynamic scattering region, then we can perform what's called a short time Fourier transform, and we can extract out an experimental absorption curve for the blood within that point. And so we can go point by point, selecting different positions. In this case, it's an artery. And we build up all of these experimental absorption curves, and we can fit to these absorption curves using sort of the oxygenated and deoxygenated curves that I showed earlier. And depending on how strong each component of the oxygenated or deoxygenated hemoglobin is in the fitting, we can quantify how much oxygen saturation is in the blood. So if we go through several different points along this artery, we can find that the mean arterial saturation is 98%, which is, I think, pretty reasonable. And then we can do the same thing for a vein, and we get a much lower oxygen saturation, which makes sense. Because the oxygen in the blood should be deposited as it goes through the capillaries and is transported back to the veins. So we don't have to manually select all of these different points along the arteries and the veins. We can also automate the whole process using some thresholding and other processing. And we can get a really nice saturation map shown in the bottom right here. And if you look, again, you have an artery and a vein, where the artery is a higher saturation than the vein. So another thing which I haven't really talked about yet is optical coherence tomography can also get information about the speed of the blood flow, which is very important for quantifying any sort of metabolic marker. So we can get this blood flow using a well-developed Doppler technique, very similar to the ones in ultrasound, or we can use a newer decorrelation technique to quantify the flow speed. And so one point I want to stress here is that in a single data set, we're able to acquire an angiogram, a saturation map, and also a flow speed map. And because they're all in one data set and all acquired at the same time, they're all easily co-registered. You don't have this problem where you have two data sets that you need to somehow combine. You don't have any sort of weird temporal offsets from doing one scan followed by another scan. And I think that's a very important aspect of OCT metabolic imaging. And so what can we actually do with this? We can combine all of these different information, and we can put it into this formula, and we can figure out a quantifiable measure of the, in this case, it's the metabolic rate of oxygen consumption in the brain. And if we look at this measure that we have here, 316 micromole per 100 gram per minute, this is in agreement with the literature, which I think is a very exciting result. And so, just a couple conclusions. I also want to mention that all of this is done without any sort of extra contrast agent. This is using purely endogenous contrast, which is another, I think, very important feature of OCT. So just a couple conclusions. We can resolve OCT measurements on the micron scale. The OCT measurements that we get in the visible spectrum can provide an angiogram, oxygen saturation, and blood flow measurements. And using all of these combined, we can create, we can quantify the amount of oxygen metabolized in any region of tissue. And obviously, this is an important biomarker for understanding a lot of diseases, and so it has quite a few applications. I mean, you can apply this to any sort of tissue, but a couple things that we think are pretty interesting is you could use it for a small animal Alzheimer's model in the brain, or while you can't do human imaging in the brain because of how invasive it is, you can do imaging in the human eye. So you can do metabolic imaging of eye disease in humans in vivo. And so I just want to acknowledge some of our funding sources: the National Institutes of Health, American Heart Association, and the Glaucoma Research Foundation. I want to thank my PI, Dr. Srinivasan, and all of my great lab mates, without whom I wouldn't have any of this data. So thank you.
We have time for questions, if anybody has.
That's really very pretty, but I'm somewhat confused. Can you compare and contrast this with photoacoustic imaging?
So photoacoustic imaging can also quantify oxygen saturation in the brain. I'm afraid I don't know too much about it. So I guess one of the advantages, I think, of using OCT over photoacoustic is, in order to get these measurements for photoacoustic, you need multiple pulsed lasers, which I think can be a very expensive and intensive undertaking. With OCT, you really just need one laser, and you can acquire using,
Can this be applied to the circulating tumor cells in the blood?
I hadn't actually thought of that application. OCT is fiber optic technology. So theoretically, you could design some kind of a probe, perhaps, to get the light near these cells that you're interested in. I think you'd have to do some other logistical. You'd have to think about how you want to set it up, but I think it could be possible if you design the right kind of probe, or if you could somehow get the tumor cells out if you wanted to look at them there.
two questions. One is, what is the trade-off in penetration depth that you get when going from infrared to visible?
Okay, so in infrared, so if the conditions are right, you can get between maybe 1 millimeter and at the very far end, 2 millimeters of depth in tissue. I'd say conservatively, using visible light, we're looking on the order of like, I don't know, 3 to 600 micron.
And the second part is, from your plots of hemoglobin, oxy and deoxy, it looks like the absorption is stronger, but the differences seem smaller between oxy and deoxy hemoglobin. I guess it's not intuitive to me that that would give you a better signal to noise. Could you maybe talk?
Okay, yeah. So the contrast doesn't necessarily come from, I mean, one component of contrast is certainly how different the, so I'm assuming you're talking about the difference here.
Right, because that looks big, that seems easy to measure, right? So, yeah.
So that is definitely, that's why they use these wavelengths in some other commercial applications. So that's only one component of what creates the contrast. Another is, so with OCT, we're looking at very short path lengths through the tissue and through the blood. So we actually want high attenuation in order to be able to see these differences. So if we use the lower attenuation, it might be harder to see any differences. And then the other side of why we want to use visible light is these sort of, these features, the 2 peaks here and the 1 peak here, can be easier to model in some ways because they're so different in structure.
I think that's all the time we have for questions. Thank you. All right.
I'm happy to present the full gender spectrum here, no pun intended. So I'm a 4th year biomedical engineering grad student, and like the young lady who introduced me said, I work for Dr. Crystal Ricklinger, and she's in the Department of Pharmacology, but in the BME graduate group. And I'm really excited to talk to you about my project, which, as the title suggests, is about integrated molecular and functional imaging of electrophysiology and inflammation following myocardial infarction. And I guess this was it?
Yeah, you're
Okay.
Sorry.
Okay, so myocardial infarction, MI, or a heart attack, in its most simple definition, is just blockade of blood supply that leads to damaged or dead myocardium. It usually happens in the coronary artery system, and we know from literature that long-term chronic remodeling can negatively impact left ventricular pump function. We also know that long-term chronic electrophysiological remodeling can alter action potential properties and conduction properties, which can lead to lethal abnormal heart rhythms known as arrhythmias. So, we know the major underlying cause of MI is atherosclerosis or coronary artery disease. It's a chronic systemic inflammatory disease state, and several papers have shown that atherosclerotic mouse models have an altered inflammatory response post-MI. So just a couple of examples of what that means is that they have increased myocardial inflammation due to increased recruitment and accumulation of inflammatory cells. Specifically here, what's important to us is monocyte-derived macrophages to the heart post-MI, which leads to increased protease activity in the infarct and peri-surrounding region. So that led us to ask this question: how does inflammation, or how might inflammation play a role in electrophysiological remodeling following MI? So we think that the macrophage is the key and critical player in the maintenance and progression of the inflammatory response post-MI. So it can secrete several proteases and cytokines that can alter cardiac cellular processes. I just have a couple of examples here. It can secrete matrix metalloproteinases, MMPs, like MMP7, MMP9, that can lead to gap junction uncoupling. It can also secrete cytokines like interleukins. Specifically here, we're concerned with interleukin-1 beta, which can lead to gap junction degradation. And for us, the important gap junction that we're looking at is a gap junction connexin 43. And this is integral to cell communication and integral to proper conduction. So if we have gap junction loss or gap junction uncoupling, it can lead to negative electrophysiological remodeling, which can lead to conduction heterogeneity and lead to those nasty abnormal heart rhythms. So based on all this information and all these previous studies and the literature, our hypothesis is that the heightened inflammatory response in atherosclerosis will exacerbate electrophysiological remodeling following myocardial infarction. So how do we look at that? Well, we look at that using optical approaches. So we look at electrophysiology using functional imaging, using optical mapping, where we stain the epicardial surface of the left ventricle with a voltage-sensitive dye that allows us to look at action potential properties and other electrophysiological parameters. We also try to correlate that with molecular imaging of inflammation, where we use an activatable protease fluorescent agent that only becomes optically active once it's been cleaved by a specific series of inflammatory cytokines, secreted predominantly by the inflammatory macrophage. Okay, so here's just an overview of our protocol. So we have two different animals that we're using. We have a wild-type animal and a apolipoprotein E deficient animal, which is our atherosclerotic animal, on a high-fat Western diet. First day, we create our infarct by ligating the left anterior coronary descending artery. Then on day 4, we'll inject our inject our imaging agent to track inflammation. On day 5, we'll sacrifice the animal, rapidly excise the heart, perform ex vivo studies looking at inflammation and electrophysiology. Additionally, we have a subset of animals that are wild type that receive daily injections after infarct up until the day of sacrifice of lipopolysaccharide, which classically activates the inflammatory response. So here's just some examples of what the data looks like. Here's our molecular imaging of inflammation, where the areas in red just indicate high areas of inflammation, fluorescence intensity. And here are our functional data of electrophysiology. We can generate activation maps, which look at how the action potential propagates across that epicardial surface. You can see here by the crowding of these isochrones that the action potential propagation is slowed down, which is not normal. It should be uniform. We can also look at action potential duration, how long the action potential is, and how long it takes the action potential to reach its maximum peak with activation rested. We can also take a look at optical action potential morphology. So what are the results? Okay, so the important thing to look at here is, here are our wild type and our operation, control animal, so they don't receive the ligation. Here you can see that they look pretty similar, but they're markedly different from our inflammatory models, this being the atherosclerotic model and our LPS inflamed induced model. And you can see that there's clear changes in activation, slowing here in these inflammatory models, action potential duration, and activation rise time. Here in the red box here indicates where we quantify the data. So here's what that quantification looks like. Also, you see here prolongation in APD and action potential rise time. You also see clear marked differences in action potential morphology between the wild type and the control versus the inflammatory models. Okay, but what about the inflammation? Okay, so here we can also see that the control and the wild type look pretty similar in comparison with the inflammatory models that have markedly increased inflammation intensity. Here we can look at 2 millimeter sections indicated by the white dotted lines. We can also see similar trends. Here in the white boxes is the quantification of that inflammation intensity. We can also see a nice trend here where increase in inflammation leads to decreasing in conduction velocity, so slowing of conduction. Okay, so then we wanted to look at what could be a possible molecular reason behind some of these changes that we're seeing. So we decided to do some immunohistochemistry, looking at connexin 43, that integral gap junction protein, and CD68, a marker of macrophages. So you can see in the remote region in the inflamed model, a remote region means above the ligation site, far from the infarct area. You can see a nice uniform distribution and normal expression of connexin 43 with minimal macrophage activity. You can see from the blown up inlet, nice connexin, no macrophages. Even absent of ischemia, the introduction of inflammatory activity in macrophages, you start to see some degradation of the connexin. This only becomes further exacerbated in our inflammatory model. We also wanted to quantify this. So we did some Western blots, which anyone who's done a Western blot, I have a lot of respect for. That is not easy to actually do it and have it work. I spent 8 hours in the cold room and I literally got a cold. But if we look at the Western blots in panel A, we can, if you take a look at these columns, if you've never looked at a Western blot, this column here is a marker column for size. Starts at 250, goes down to about 25. There are 3 just to make sure we did it right. Here in these columns is our wild type remote, wild type infarct region, ApoE remote, ApoE infarct region, and this is, just ignore this column for now. In this column here is an important control HEC cell lysate, which should not show up for any of the markers that we're looking at. So in this, we can see here that in the ApoE animals, you'll see elevated levels of interleukin-1 beta. Here, in looking at connexin 43, you'll see a decrease and a shift, which is, we think, is because connexin is being hyperphosphorylated in the inflammatory models. But if you really want to know more about that, you can ask me about that later. So we see a nice trend here with increasing expression of interleukin-1 beta and decreased connexin levels in the infarct region versus the remote regions. Okay, so we made them sick and we made them fat, but can we make them better? And being that my PI is in the Department of Pharmacology, I did a pilot study looking at a compound developed here by Dr. Heiko Wolf in Department of Pharmacology that specifically targets and inactivates macrophages among other inflammatory cells. So it's a novel anti-inflammatory. So if you ignore all this, this was the pilot study and we thought, yay! We reduced inflammation, but then we did a full study and we got mixed reviews here. We have some reasons why we think that happened, but we're still working on applying novel anti-inflammatories. And I'm going to skip the summary slide for the sake of time and go straight to the acknowledgement slide. So obviously I did not do this work alone. My PI has been a great mentor to me. Dr. Lingguo Wang did all the surgeries and helped me with majority, like all the optical mapping, pretty much everything in my project. We used to have a postdoc in the lab who's a great help to me, and my mentor in Department of Pharmacology, Dr. Don Bruce. I'll take questions.
Just one very technical question. When you're imaging the heart from a single point of view from the top, and is that what you're doing for some of the? And it's curving down to the side. How do you adjust for the geometry of what you're imaging?
So for the optical mapping, we don't. We know that there's going to be loss there. So what we do is normalize all the signals to each other to compensate for that curvature. For the epifluorescence imaging, we select ROI regions that are not below that curvature. So we know that the intensity is not uniform. Perk and Elmore will tell you that. It's 80% uniformity, and we know that there's drop at the edges. So what we try to do is center the heart, and specifically the left ventricle, as close to the center of the illumination field of view as possible, and then try to select RLIs within that region.
What was the dosing regimen with TRAM34?
Yeah, well, so the TRAM34 was a pilot study, but the dosing regimen we did twice a day, so at 12-hour time points, 40 milligrams per kilogram.
And good afternoon, everybody. Today I'll be sharing with you some very recent, very exciting, I think, results where we applied simultaneous PET and MR imaging to a mouse model of stroke. So this information is probably very familiar to most of you, but I would like to reiterate some of the points about the advantages and disadvantages of MR and PET. With MRI, of course, you can image down to very fine, refined structures at the sub-millimeter level. And there's excellent soft tissue contrast, but it does suffer from low sensitivity, although we saw that this morning that point is being addressed. PET, on the other hand, offers very high sensitivity, down to the picomolar concentration level, but it does suffer from lower spatial resolution. So, as you can imagine, being able to combine these modalities, that could be very advantageous. And this has been done. In the past decade or so, the clinical and preclinical systems have become commercially available. The early ones were sequential, but more recently, the simultaneous systems have become available, as we saw earlier. Now, on the commercial side of things, there hasn't been really any system dedicated to preclinical imaging, so that's where the Cherry Lab comes in. The lab has some of the world's leading experts in PET hardware development, and we have this latest generation PET insert that we use to collect data that I'll be describing in this talk. And basically it sits inside a Bruker 7 Tesla magnet with the RF coil going inside of the insert. And so a question that comes up is why image simultaneously rather than sequentially or even on two separate systems? And I've listed just two of the advantages here. Of course, you have improved spatial co-registration in some cases, but of course the important point is that you have temporal co-registration, being able to acquire different kinds of data sets from a biological process at the same time as it's occurring. A really good example of this was in a recent paper from a group in Germany. They basically showed changes in blood oxygenation and also FDG uptake in a rat whisker stimulation exercise. And so I think this is a really neat example of the potential applications for simultaneous PET/MR imaging. So there are also applications to stroke, and stroke is, of course, the injury that results from a loss of blood supply to the brain, and it's the second most common cause of death and a very big cause of disability worldwide. And it's highly dynamic in its nature, and by that I mean the physiological, environmental, molecular events that are occurring in the brain happen very rapidly and also are very complex, making this disease or this injury very difficult to treat. One subtype of stroke is known as cerebral hypoxic ischemic stroke, and that's a major cause of disability in children. There's actually, the incidence is as high as 4% in preterm births, and this can lead to major developmental disorders later in life. And current options for treatment still have room for improvement. There are animal models for studying this type of stroke, and I'll walk you through an example here. Essentially, you just tie off one carotid artery. This in itself is not a problem because there's a lot of redundant vasculature in the brain. But when you subject the animal to low oxygen conditions or hypoxia, this causes a rebalancing of the blood distribution in the brain, and that then leads to the ischemic stroke. Thus, you have the hypoxic ischemic stroke in the animal. And literature has shown that indeed the physiological changes that are occurring in the brain happen as soon as you start the hypoxic challenge. So in this example, they showed with laser Doppler flowmetry that there are changes in the cerebral blood flow immediately, and they also showed in a separate group of animals using MRI changes in the diffusion and also cerebral blood flow before, during, and after the hypoxic challenge. So this is a really good example of where you might be able to apply simultaneous PET MRI imaging. Precisely because we can now acquire or observe the dynamic changes in the brain as they are occurring during the injury. And in the following slides, I'll show some examples where we image with diffusion-weighted imaging, diffusion-weighted MR, and also FDG PET. And I want to emphasize that because this injury is pretty much a one-time, one-shot deal, in order to image this real-time in one animal, you really do need simultaneous imaging. And so here's the workflow. It's fairly self-explanatory, but we first acquire some MR baseline, and also we begin the PET scan, which is a constant infusion of FDG. And then we start the 30-minute hypoxic challenge, which is 8% oxygen, all the while acquiring diffusion-weighted images and also PET data, and then of course a little bit afterwards as well. And here's a picture of the hypoxia setup. Essentially, we just feed this into the breathing tube for the animal as it's on the scanner. I've also shown the coil here. It's nothing fancy. It's just a volume transmitted receive coil that's actually kind of poor in performance. But, so here are the acquisition parameters. And we used an EPI-based diffusion-weighted imaging here, which has some drawbacks, but we wanted to have the fast speed so that we can acquire, within that 30 minutes of hypoxia, at least 2 images. And as you can see here, this is an example of the PET in the background and the overlay of the volume of acquisition for the MR data. And then now some results from the MR imaging. These are ADC maps of the apparent diffusion coefficient. The lower on the color scale, the lower the diffusion, which is an indicator of cytotoxic edema, which is a hallmark of early stroke. And then we also, this is another way to look at it. These are, on the right, you have the 9 animals, tracking them over the different periods, and on the left is the average of these values. So as you can see, the trend is fairly robust, and this is actually what we expected with this animal model in terms of their diffusion changes. And so now for some PET data. This is an example here of the constant infusion. As you can see, there's an area of low FDG uptake, if you look carefully on the right, in the brain. And that is suggestive of lower glucose utilization in this animal. Here I've shown the time activity curve of FDG uptake for this animal. And in 2 other animals, we observed in one of them the same trend. And in mouse 7 and 9, so that's this blue and red curves, they also showed evidence of changing diffusion indicative of cytotoxic edema. So with 3 animals, it's still kind of early to make any conclusions, but I think what this suggests is, or demonstrates, is the utility of simultaneous NMR imaging in individual animals where the evolution of the progress of a disease or injury are different from animal to animal. So, in conclusion, I think we've demonstrated an interesting application of simultaneous NMR imaging, and we observed significant changes using MR and in 2 or 3 cases using PET. There is still quite a bit of work, but I think that it's promising and opens some, or demonstrates possibilities. One of the examples is that we're potentially looking at seeing the predictive value of NMR in response to therapeutic intervention in stroke. And I think going forward, this question, what interesting questions require and benefit greatly from simultaneous imaging, is going to be very important, because for a lot of applications, you really don't need simultaneous imaging. But I think that, well, I hope that today I've shown you one example of where it might be potentially very useful. And with that, I'd like to thank everyone listed here. Without them, it would not have been possible. And thank you for your attention. Yes.
So you listed the advantages of simultaneous imaging. Are there any disadvantages to using both at the same time?
There can be, and I think we saw some of the hardware challenges earlier. But I think the advantages outweigh them if you can address those challenges. And I think Martin back there knows quite a bit about the disadvantages. But they're mostly about implementation.
Okay.
On one of your plots you showed, the one with the 9 animals, it looked like you had some outliers that didn't follow the trend. Do you have any inkling as to what was going on there? Or?
Right. Well, there's several animals that have this issue. This one I don't really know about. The ones that ended, basically they died at this point. This one is just my error. I didn't acquire the right data this time, but I think it kind of shows an example of why simultaneous imaging could be important, because you're able to track individual animals on a case-by-case basis. And whereas if you were to do it sequentially or in separate groups of animals, you have averages that tell you these longitudinal changes.